ओके सो हेलो एवरीवन, आज हम स्टार्ट करने वाले हैं फ्रॉम द वेरी फर्स्ट चैप्टर ऑफ बायोलॉजी, स्ट्रक्चर ऑफ क्रोमोसोम, सेल साइकिल एंड सेल डिवीजन। तो आप यहाँ पे देख सकते हैं कि इट इज़ रिटन द सेकंड चैप्टर। जो फर्स्ट चैप्टर हमारा है इज़ बेस्ड ऑन द स्ट्रक्चरल the structure and the functional unit of cell. What you need to do is go through the first chapter because that's not in your syllabus, okay? But you need to know such certain terms such as cell membrane, cytoplasm, what is nucleus, what is cell fluid. You need to know such terms. So that you know such terms, the chapter is made. It is basically the revision of class 9, okay? Cell was also there in class 9. So let's get started with the second chapter of biology, structure of chromosome, cell cycle and cell division. Okay, from the very first paragraph you get to know how cell division is very very important in our life. How from this very first living being, or say a small ant, to the very big elephant, the cell division plays a very important role. Cell division is used for growth, reproduction, stimulation of regrowth of cells. So thereby we'll get to know further in the chapter how cell division is so important in our daily life. And moving on to the very very important paragraph of our biology chapter, this chapter, what are chromosomes? First of all, going on to this paragraph, I don't seem to see a very good flow in this paragraph. So I have made some notes which you have to see and I'll teach you. Okay, what happened previously is I forgot to switch on the video option of the desktop, thereby I have to re-record it again. So in the I have also written the syllabus at the top, that what is basically there in this chapter you need to know. First of all, we are going to finish this, then we are going to move on this, because if you don't know the structure of chromosomes and the complete flow of DNA, structure of whatever, you will not get to understand the cell cycle and cell division. First you need to know the structure of chromosome. So what happens is, you often have seen the structure of DNA. You're well known to the structure. In every social media platform you have seen something like that. DNA is also known as macromolecule and it has a double helix structure. Okay, that is what I have written in here. You'll get the notes, no need to panic. What happens is that the DNA opens up and it wraps onto such 8 histone proteins and forms a histone octamer. Now, what are histone proteins? What is an octamer? Let me explain to you very very precisely. What happens? See, histone proteins are histones are basically particular type of proteins which help in the wrapping and packaging of DNA into a histone octamer, or also known as a nucleosome. See, suppose this is the DNA strand. Okay, this is the DNA strand. It is opening and it wraps onto such 8 histone proteins. Okay, I'll make 8 histone proteins. 1, 2, 3, 4, 5, 6, 7 and 8. Okay, 8 histone proteins. It wraps onto that like this, and this total thing, okay, these things also happen further. ऐसे ही होती जाती हैं further, ऐसे और है, ठीक है, मैं नहीं बना रही, वो अलग बात है. This total thing, the histone protein and the DNA as a whole, as a unit, is known as nucleosome or a histone octamer. See, very very important. It is often asked in the question, what is a nucleosome or what is a histone octamer? We will write that when the DNA wraps onto 8 histone octamers, it forms into a histone octamer or a nucleosome. Now further, such units are formed. They further get coiled. Into a colored like thread structure known as chromatin, which further gets coiled into chromatid. Now what happens to the chromatid? You see the flow. DNA wraps onto 8 histone proteins and forms a nucleosome. The nucleosome further forms, it gets coiled up and form a colored thread like structure known as chromatin. The chromatin further coils up into chromatid, which further coils and loops into 2 sister chromatids and joins at the centromere. Now what are the sister chromatids? See, this is one sister chromatid, this is another sister chromatid which joins in the center. You see the black dot I have made, and this is known as centromere. Very, very, very important question. What is a centromere? And this as a whole, I mean the two sister chromatids joined at the centromere form a total chromosome. And this, my dear, is the development, or you can say the basic structure of chromosome and how it is formed with the flow. DNA plus histone proteins, then the nucleosome, and then chromatin to chromatid, then to a sister chromatid joined at the centromere and forms a chromosome. I hope you have understood up to this that what is a chromosome. I will personally request you to go through this paragraph. It is very, very easy paragraph. You need to go through this paragraph and always add this thing in your answers whenever it is asked what is chromosome. Or you can even write in the flow I have written. No teacher can cut in the flow I have written. Okay, I can assure you that. Go through this paragraph. The important line I have underlined, but you need to go through this paragraph. Read this paragraph. It is very, very important, and this structure as well. This thing is so important that you can even imagine. Okay, this itself is a very big question, and it is very, very, very important. Okay, moving on to the discovery. No need to read the discovery book. Waste of time. See, in this diagram, see the chromatin fibers coils up into chromatid, which further coils into chromosome, which has two sister chromatids joined in the center. The center is known as centromere. Moving on to chromatin. Chromatin means this thread-like structure. This thread-like structure is known as chromatin. Now see, what is chromatin made of? Chromatin is made of DNA and histones. DNA and histones together form nucleosome, which further gets coiled into chromatid. Got to know how the flow was. So what is you need to know in this paragraph is the full form of DNA is deoxyribonucleic acid, and it is about 40% in chromatin, and histone proteins are present of 60%. So it is more present in the chromatin. The histone protein overrule, because there are 8 histone proteins in a nucleosome, so it always has to overrule. See, a single DNA molecule is very large and hence is described as a macromolecule. See, very, very important. Composed of 2 complementary strands, each other in a bond each other in a double helix. So this is a short question. What is the structure of DNA? It is a double helix structure. Moving on to this diagram. First, you need to know what does a DNA consist of. The DNA I need to move this thing up. So what happens is the DNA is mainly composed of 3 things: NPP, nitrogenous base, pentose sugar, and phosphate group. NPP. This double P, that means the pentose sugar and the phosphate group together form nucleotides. This is a short question, that what are nucleotides and what are the different types of nitrogenous base: adenine, thymine, cytosine, and guanine. Next, you will get to know how adenine and thymine connect with each other with 2 hydrogen bonds, and guanine and cytosine bond with 3 hydrogen bonds. Okay. See, it's written in here that the nucleotides, which are made up of 3 complement components, phosphate, sugar, and nitrogenous base, which are attached to the sugar inwards, that extend to join by a hydrogen bond of the complementary nitrogen base from the other strand. Thus, the 2 strands together make a ladder-like arrangement with the nitrogenous base forming rungs of the ladder. Rungs. What is the meaning of rungs? I mean, you have seen a ladder, right? The part where we put our foot to get up of the stairs are known as rungs. Okay. 4 types of nitrogenous base. Important question. A, G, C, T. Adenine, guanine, cytosine, thymine. Adenine pairs with thymine with 2 hydrogen bonds, while guanine pairs with cytosine with 3 hydrogen bonds. You can see in here. Okay, go through this diagram as well, because in schools they are often asked that such a simple similar diagram is given and you need to identify what is phosphate, what is sugar. It will be very childish kind of question. No need to worry about. I will also bring a series of the important question discussed chapter wise, so you'll get everything in this channel. Okay. Histone proteins. Histones are the protein that help in the coiling and packaging of DNA into structural units called nucleosome. We have already read that. But this is nucleosome. See, so that the thing is that in this whole 2 pages, the flow isn't that well good. That's the reason I first explained you the whole flow, and then I am going as such is given in the book. So the way I teach, I hope you find it easier. Now the next line, the DNA strand winds into a core of 8 histone proteins called the histone octamer. Each such complex is known as nucleosome. We already read that. An individual nucleosome can be imagined like a football around which a long rope is wound with 1 or 2 loops. It means, say such, this is a football. Okay, don't curse me. Okay, this is a football. Just think that this is a football. This is the DNA strand which winds upon this thing with 2 or 3 loops, and this total complex is supposed to be known as nucleosome. This is a real life example given, but in here the football is the 8 histone proteins, and the rope that is wrapped around it is the DNA strand. Okay, a single human chromosome may have about 1 million nucleosomes. Okay, not that important. Now, what is the actual size of a DNA? I mean, the whole length is 2 meters, but it is if stretched end to end. But in our body, so that it gets fitted in our nucleus, it has to get coiled up so that it adjusts itself in the nucleus. The nucleus of a human body is only about 6 μm in diameter. Thereby, the DNA needs to be condensed in order to fit inside the nucleus. The entire chromatin fiber is coiled and supercoiled, something like the coils and supercoils we see in a typical telephone cord to eventually form nucleosome. It is a real life example, like the same application we see in telephone wires, that the wire itself is very long but it is super coiled so that it gets adjusted up. Okay, given below is the whole flow that I have explained in the diagram is given in here. Go through this, is very important. This is the cell and this is the nucleus, and in which, see, this is the whole total nucleus which is zoomed in and shown that so many chromosomes are in the nucleus, and they get adjusted inside it, although they are made up of DNA. Structure of chromosome, we have already read how the chromosome, there are 2 sister chromatins attached at the point of contact, and the point of attachment is known as centromere, and it appears to be a small constricted part. Small constricted part means in which the part it's connected, see, the part is a little slimmer, so it is a constricted part. सिकुड़ गया है थोड़ा. Now moving on, this particular side is known as centromere. The centromere also serves as the attachment. Okay, in this thing, just go through this, but here this thing you will read later in this chapter when you come to the see this whole phases. I mean the resting phase, the anaphase, metaphase. That phases you will get to know how the centromere is the main part from where the sister chromatids again deattaches and forms other things. Okay. So I'll request you to read this paragraph, go through the paragraph, but you will not understand, I think. But not right now. As we move on, you'll get to understand. Okay, take a break in here, pause the video, take a break, read the whole chapter until I also drink some water. Just a minute, let me also pause it.